Yeah, they can then also kill cells which should not die. That's why you have in Alzheimer a lot of brain cells dead, without which you suffer and die, or amyotrophic lateral sclerosis or Parkinson. There's a lot of these similar diseases where cells disappear despite they shouldn't. So it's like the other side of the coin. And we are chemists or physical chemists. So we come more or less like from the physical chemical side. So we are not interested in the whole big bodies, on what goes wrong with the patients, on the molecular level, what goes down at the cellular level. But to come down to this part, we have to think about what is controlling programmed cell death. And we have two major pathways. We have an extrinsic pathway, where you have signals outside the cell who trigger some receptors, and then inside the cell they start activating caspases and so on. But you also have an intrinsic one, which is coupled to your power station of the cell, this mitochondria who make ATP and all. And at the end, it's destroying the mitochondria, making pores, also activating caspases, and finally destroying the cell. So then the key is quite simply, we say you switch off the powerhouse of the cell, the mitochondria, the cells will not make it. And both pathways are actually connected to each other. They're not completely independent. But the intrinsic pathway you get very nicely by high intracellular stress. For example, also a lot of cancer drugs. They induce high cellular stress to induce cancer cell death. And sometimes the cancer cells, they produce so much survival proteins at the mitochondrial surface, like BCL2, that they escape this triggered cell death. But we'll come to this a little later. So what happened now on the mitochondrial level? If you look at mitochondrion in your cell, has an inner and an outer membrane. In a simple way, if you want to kill mitochondria, you use proteins which make a hole into the outer membrane, like here shown. Then cytochrome C and other molecules can come out and they trigger this cell death. It's like a signal cascade. The caspases get activated and so on until mitochondria are dead, cell dead, and so on. So at the end, you just make a hole and cell dies. And this mechanism is very tightly controlled, that normally you don't have a hole in your mitochondria and your cells look happily. And you have a big family, it's called BCL2 family. It's like B cell lymphoma 2 family, because it was discovered in a cancer of a patient in a gene, and this gene is called BCL2 itself. And this was actually a so-called survival or anti-apoptotic protein, because it got overproduced in many cancers to protect them from getting killed. Just like if you say you want to go in through a door and you have a lot of bodyguards who do not let you in. Then more people want to go in and the cell produces more bodyguards. So the drugs or something, a signal never makes it in. Little like this. You have tons of bodyguards, so the death signal never makes it through the door and never makes a hole. It's a whole family. BCLXL is the other one and a lot. But then you also have the opposite side. You have quite similar structures, but there's a cell killing or apoptotic ones like bugs, and they make the pores. They bind to the mitochondrial surface, make a pore and kill it. So we have now a working model from our physical chemistry stuff and also from a lot of literature and work. What is our working model is like we have a mitochondrial outer membrane, which is also complex lipids. Then we have BCL2 sitting somewhere on the membrane, like a bodyguard. And if Bugs signal comes to kill the cell, normally BCL2 can neutralize it. And if you say a cell should get killed, you get a lot of Bugs, which will neutralize and make pores, make a hole, kill the cell. And you can always think about in a cancer cell, which has a lot of BCL2, it will always neutralize all Bugs which is coming. So you never make it to this pore. You will always neutralize it and then the cancer cell happily survives. But from the physical chemistry point or structural biology point, what is quite important is that the BCL2 location in the membrane is not known. It's not known if it's sticking out like a peripheral membrane protein, integral membrane protein, like rhodopsin, what it's doing. The structure of the full BCL2 is not known because it's an insoluble membrane protein. And what would be the holy grail in the research is like this complex is not known. And this complex you have normally in all your happy cells. If any bugs comes, it's neutralized, so your normal cells are happily alive because they both neutralize each other. We thought, and this is like all these questions which we work on it. Membrane, BCL2, and so on. We thought it was quite important. This was for a long time. I come from the lipid field. It was for a long time ignored a little, you need quite good proteins. So the problem is not to work with the membrane part. The problem is to get fully functional proteins expressed and in milligram amounts, both. And this was for a long time a big problem, which is now one of the postdocs, Jürgen, solved quite nicely. But before we come to this, we got in a short backflash into what are membranes. We thought the membranes are lipid bilayers with proteins and peptides and sterols and so on. We thought, and you also need for working with mitochondrial membranes, you have to try to make a quite realistic model. Also not only use one lipid and try to use the lipids they use there. Without a membrane, you have something which is functional, a little related, and not only a model membrane, which is so nice to handle, but has not much relevance. And so what is the advantage with lipids? You can buy them a lot, isolated. It's like DMPC. So my whole PhD thesis was made on DMPC relaxation with NMR. If you put them in water or buffers, they automatically make bilayers spontaneously. So you have automatically a nice bilayer, which is like in the real cell membrane, the plasma membrane, and all the other organelles. So it's a very nice, simple system. To make it a little more complicated, you can make like unsaturated lipid, palmitoyl phosphatidylcholine. And if you say you want to simulate oxidative stress in a membrane, you put some oxidized lipids in. Here's PACE with a carboxyl group at the end of fatty acid chain, the aldehyde. We thought, and what we use for mitochondrial membrane, we use PC, PE, gadolipin, and sometimes also oxidized lipids. So we use a little quite nice ratio where we try to realistically mimic a little the mitochondrial outer membrane. It's not perfect because real membranes are asymmetric, and we make it symmetric, and they have some more, they have a little sterol and so on, but the basic functions they all have. And what is nice with the lipids, if you also can do solid state NMR, I only show example. This is like shown here. It's like a carbon-13 solid state magic angle spinning NMR spectrum, carbon-13, direct polarization. You see all carbons. You can make a fingerprint with your NMR machine where you can look at all different carbons in your membranes. You have like atomic resolution because you see each, the ester bond, the vinylene, glycerol, like the choline group, which would be up here. So you see everything. And you can look, if you put something into the membrane, protein or anything, how different parts are changing. So you can look if something is changing here or here or here. But it has a huge advantage. It's a non-invasive method. You make your sample. You don't need any special label. It's natural carbon-13 abundance. You don't need any fluorescence label. You need nothing. Put it in and run it. So it's a quite nice thing. And you can assign models in most groups and look what happens. You can do the same with phosphor NMR or with proton, so you get quite a lot of information about different atoms in your bilayer and what will happen if you change something. Okay, but now we go a little more to the other side. But before we go this, you can also look calorimetry, so you can look also for the basic phase behavior of this mitochondrial membrane. What happens if you change temperature, put oxidized lipids in? And one of these profiles is shown here. And you see quite nicely, if you say it's a heat capacity, it's like melting ice. Melting ice, you would probably have it at 0 degree melting, and here the lipids go at higher temperature. And you see it's not ideal, but you see POPC with gadolipin is like 293. And if you have oxidized lipids, you have even much more complicated melting curves. If you would have pure ice, you would have a sharp line at 0 degrees Celsius. And here you see this is quite non-cooperative, quite broad phase transitions in these complex membranes, which is means they are quite disordered and not really ideal mixing. But if we say now we need the other side of our system, we need also proteins. We thought this is Jürgen. He works in my group for a long time. He makes all these proteins. And he more or less developed the protocol to get huge amounts of this BCL2 out. Because BCL2 is a non-soluble membrane protein where people for many years had to make a soluble variant where they put the transmembrane part away and a loop and chimeric system to get it soluble for some NMR, but they never figured out how to make the full-length non-soluble membrane protein. So what we have to do when we purify it, it has to be in detergent, because the membrane proteins without detergent they aggregate and you don't have anything. So it's clear if you purify it, you put in detergent, and then if you want them in a membrane, you have to do reconstitution back in a bilayer. And it's like here DMPC liposomes. You see here, if you make a density gradient, you can centrifugate it back or reconstitute it back in its membrane similar environment, which is quite important. And this works quite well. And now we have to think about, okay, now we have our membranes, we have our proteins. What can we do now? Now comes the title of my talk. We can do neutron reflectometry. So we can see an overall picture. How is the whole membrane organized with proteins on top or inside? This you cannot do with NMR. NMR, you have an angstrom resolution, but you cannot say what is 500 angstroms away or 100 or something. So it gives you a global overall picture, what happens. And then you can use NMR, you get a molecular picture. You get like atomic detail information, how is the structure, how is the residues, how is the lipids behaving. So you get quite a lot out. And so on what we were doing in the research, we were looking BCL2, how is it sitting in the membrane, on top or in or what. We can look at bugs, if it's sitting on top or inside. And we can look, this was this third question, what happens if BCL2 and bugs form a complex? How is this sitting in the membrane? Because this is a fundamental one where also a lot of drug companies try to make drugs which dissociate this part, that bugs can make a pore in the membrane and kill a cancer cell. This is like Epi and a lot of companies develop. And there's also one drug on the market, venetoclax, but it works only for chronic leukemia, unfortunately, and not for so much other things. Here comes now NMR spectrum. It's HSGSU with N15 labeled protein in detergent. So we use small detergent in micelles as a membrane similar environment to run solution NMR, which has a much better resolution than the solid state, to get information about our protein. And what you see quite nicely, you see a lot of peaks, a lot of dots, and each N15 dot is a residue. And we see around 190 in total amino acids of the 239. But what we see all glycines, which we can also assign. So we can say which dot is which glycine in your sequence. And so we can say, okay, we have a glycine at position 5, at 8, up to 237 or TM transmembrane part. And what this was another postdoc in ARMEC, we can also run NMR to look which residues are water soluble, also sitting on the surface and seeing some aqueous environment and which not. And with these clinics and the red ones, they all have contact with a buffer. All the others, the black ones sitting inside the micelles, are protected like in a bilayer, but the red ones, they can look out and have a little contact on the surface with water and are also often more flexible and often also sharper. We thought, and this is a quite nice way that you say, it's like a fingerprint. You have your 10 fingers, like we have our glycines. Then you can look for each finger or each glycine, what happens if you do something. Because if you add a ligand, other protein, will all 10 fingers see the same, or some fingers will not see anything because they're in the wrong part of the protein, and this other protein interacting only with this part. So you can use quite a lot. You can use the other residues, but here we use glycine because our glycines are distributed in all functional parts of the protein. The BH4, which is at the N-terminal part, it's intrinsic loop domain, which is like a control or regulative unit disordered. And then this is a main part, it's BH3 to BH1, it's like the binding groove BCL2 for bugs. This way here, it's neutralizing proteins which want to kill the cell. And then it has a transmembrane part to stick to the membrane nicely. So now comes important thing. If they, okay, I have a nice protein and so on, what does it mean? If you do work, you have to look at protein functions. So what we do, we use a very simple assay. We use not the full bugs, we buy peptide, which is like a so-called BH3 domain of bugs, which is like the big binding motif of bugs to BCL2. So you can buy this 36 amino acid long peptide. It's called a bugs BH3 death domain. And you titrate in this ligand and look if the protein is binding or not to it. So then you see quite nicely, you can titrate it from 1 to 1 to 1 to 12. And you see a lot of residues are changing. So they see something is binding. And the good thing is, like I told you with the 10 fingers you have, here I show now 6 fingers or residues. You can see that they also see different things in binding. So for example, here you see the chemical shift. It's called chemical shift perturbation. It's changing N15 and proton because the local environment changes if you bind something to it, or conformation or electrostatic. But you see here it's in a different way. See here it's bended. So you see also that not all part of the proteins experience a simple on-off ligand. Some might have multiple binding modes of flexibility. Some have probably very simple on-off binding mode. So this means that different parts of the proteins probably see this peptide slightly different, which is very interesting, but we have not figured out what it means. Now, this is simple, the straight lines, but this is a little more tricky. And we can also make affinities. So it's around 500 micromolar affinity how it's binding. And so we can also say, okay, this residue see quite nicely bound and so on. We thought it's a quite nice functional assay. We have then also others with the full protein, but it's always the first step to look our protein is after purification, unfolding, refolding, detergent, it's still binding, doing something as it should. Also, the circular spectroscopy looks also good. Then to test it with another method, we were doing isothermal titration telemetry, and here we use the full bugs, wild type bugs. And you see very nicely that it's also binding with 2 micromolar affinity, and we were doing before surface plasmon resonance with another detergent, but below critical micelle concentration, it's above. Here we saw it's binding with 35 nanomolar affinity. We thought, and this is also very nice. We see it's binding, but it's clear if you look at literature, it should go down to nanomolar. That if you work with detergent, it's not really like if you work in a real membrane or solution. This detergent micelle bubble, they're protected partially, they have the same behavior, but we thought, but the affinities are changed because the peptide also needs micelles, detergent, and then go to a protein with detergent. So this affinity will be not the same as in a real world. We thought, but its behavior is the same. We thought this is got discovered for a lot of membrane proteins, that in micelles, it behaves the same, but they have not the same affinity or the main features. And this one has something to think about if you work with detergent. So they say they have also limitations. They have big advantages. You can do solution NMR, you can handle, but also the affinities are relative affinities. Okay, then we look for another NMR part. This was, we look now also for the chemical shifts when you plot them for your different fingers, just like your finger glycine 5 to 237. And you see already the biggest changes. You have 141 and so on in the area where you have the binding pocket. This happens most in the flexible loop region. Here happens not much. Transmembrane part happens not much as expected, and the BH4 part will also happen some stuff. So this is also, we discussed the second regulative unit. And this is really that our system in the micelle responds like to binding as expected from former studies. The different parts see either the peptide or not. And this is a quite nice result that you say you can, with residue specific resolution, see what happens, because the structure is still not known. We're still working on it, but we do not have the structure. It's like only a kind of cartoon of the soluble BCL2 with a loop and other region connected to it. So we still have to work on it. So what do we do now? Now was a question at the beginning. We want to know what is BCL2 doing in the membrane. Is it sitting here or what? And now comes a neutron work soon. And for doing neutron work, we do neutron reflectometry. We have a single bilayer on a silica crystal. We have to reconstitute our BCL2, not only in basic bilayers, we also do it in more mitochondrial-like bilayers like POPC, POPE catalipin. So we have to work a lot also on getting protein from a detergent into a complex membrane in. So you have to work with detergent, you remove it by dialysis with biobeads, then you have to concentrate, make a density gradient centrifugation to separate your bilayer with a protein. We use often 70:1 here. And then you say you can go to a neutron source. And we thought, and we have a long collaboration, Sweden with United Kingdom on the so-called ISIS or Rutherford Appleton labs in Havel outside Oxford. Because we thought in Lund they build a new neutron source, ESS, European Spallation Source, which is not ready yet. So we do everything in Havel, or the other place would be Grenoble at ILL. So, and if you ever go to a neutron source, if you are new, you always get the oldest part. So we got like SURF in the old target station, which says no space, nothing, and had to work there. So in the basement, also nobody ever visited us in this part of the target station. And there we could work in Ruhe. This is Jürgen, then Tobias was with. So we have a control unit where you can run the neutrons and have to look for the data and tell the machine what to do. We thought, and this is like you can book 3-4 days or apply for beam time. And if you get it, then say, want you also to be 24 hours there. We thought with our old machine here and our membranes, which is not sensitive, this was very nice because we could always run quite 8 to 12 hour runs. So say you start up your run and then you go to the pub and in the morning you look what happened. But if you have other machines, can be that you have to change each 2 hours and have to split up or try to have an automatic system in. Okay, now what are the results now? That's not so simple for me. So you get like neutron intensity over the wave transfer vector, or with a neutron different wavelengths, and you do so-called isotope contrast. So you measure neutrons which are sensitive to deuterium. You measure them in protonated buffer, you measure it in deuterated buffer, you measure it in mixtures. That you say you can, for example, filter out your protein. If you have deuterated protein, you can make the protein visible or invisible, the lipids visible or invisible, or your silica crystal visible or invisible. And so you can figure out little what is each component doing. And then you have to make a so-called scattering length density profile. And here's like the membrane from the silica across the membrane. So you go from 0 up here. Like you walk through the membrane and see the densities. So you see the inner heat groups of the lipid, fatty acid tails, the outer, and then is the aqueous solution, what's outside. And then you do it before and after BCL2. With BCL2, you see already something happens. This goes forward. It was deuterated BCL2. So they say BCL2 is somehow sitting where the fatty acids are sitting. So it's more sitting in the membrane. So now you can do it in a more clever way. Although not me, people at neutron source, they are quite clever. For me, this is too tricky to analyze. And when I went there to learn some of this basic analysis for student, you can do business bilayer, but then they said our system is too complicated to do themselves. So I can do their textbooks examples, but not our measurements. What you can do, you can make a volume fraction SLD plot. So you say you plot each component separately when you walk through. So you can walk more or less through your bilayer from the silica. You can say with the water, you get a little more water where the heat groups are, then you get no water where the fatty acids are, there's the brown ones. You get a lot of water if you go out, and then in the buffer above the bilayer. But important is like our purple is BCL2. you get BCl2, you sit, all the density is sitting more or less in the bilayer. It's not looking anything outside. It goes to the head group, but not more. So it's a quite embedded, inserted protein inside the membrane, not looking out much. This was very surprisingly, because from the literature before, we thought it's sticking on the surface, but how we do it, it's not. It's really sitting inside, but it has some glycine residues, which are water accessible. So at least on the sides, something must be a little have contact with water. And that's why we went back to NMR, and this is with a cross-polarization, solid state. And this is a very simple method. You look for non-mobile residues. So if you're sitting in a bilayer, you look for things which do not move, residues. You see quite a lot of residues, which are carbon-13 labeled all. They do not move, not at low temperature, not even at high temperature, only the lipids. If they melt, you see some sharp lines coming from the lipid and bilayer environment. And then you can make the opposite. This was cross-polarizations, and you make like inept. And then you say you can look only for things which are looking outside the membrane, are highly flexible, like these things. And then you see also at low temperature, it's not much flexible, because they even had to multiply it a lot. So real intensity is really less. And even if you go to 313 Kelvin, you see a couple of sharper lines, but not much. So most of the protein is not mobile as expected, sitting inside. And like the flexible loop and some glycines, they're sitting at the membrane interface with water in contact, so they're flexible and sharper. But I thought, and this small amount you cannot see by Neutron if you have 10% or so. But the NMR also supports the case that most part of the protein is sitting inside a bilayer, not doing much. Okay, then we made a model which we published in Communication Biology last year. To say our model from the Neutron is sitting inside. From the NMR and glycine, you must have a little loop which is looking outside, flexible. We thought, and you can say it in a quite nice way, the BCl2 is blocking cell-killing proteins on the membrane level. So if bugs comes in here, it cannot make a pore. But the regulation of its function comes like from the loop region, which can phosphorylate, which comes from the cytosol. The signals can come in from the cell, do something with the loop, change the function. But we thought, but the body itself, it's like a two-dimensional working protein if it works in the membrane layer. Okay, now we said we have done this. Now we do the next one. We do what happened with bugs, the protein which is making the hole in the membrane. So if you look at old NMR, it's like Arthur was doing it and Tobias a lot. If you look here at bugs in the membrane, you see a lot of flexible ones. You see also a lot of non-flexible ones. So you see quite here that part of the protein is sticking in the membrane, but a major part is also outside, but flexible. So it's behaving quite different to BCl2. And this was quite important, that it's quite different to BCl2. And if you do not remember Arthur, he thought he left us a year before COVID came and went to this company BioNTech in Mainz, which nobody knew before COVID. And now they make also vaccine for Pfizer. And now he's like responsible for all the quality control. So he made quite a good career, and because he went to the company because they want to make cancer vaccine. So he said he had the right feeling in his stomach to go a year before he got famous to this company and not to Merck. Most other people went in his home area. So, and bugs now to check that it makes hole. We make leakage assays with the people in Prague, with Martin Hoff, Sharka Radek. There's also academic science at Heirovsky Institute in Prague. They do a lot of fluorescence. And then what you do, you have calcine inside the vesicles, and then you add bugs. And you can see if you have no cardiolipin in it, not much happens. If you have cardiolipin in and oxidized lipids, a lot of things happens. So important was that you need cardiolipin in the membrane, like it's in the mitochondrial membrane naturally. And oxidized lipids both increase the attachment of bugs to the membrane and causing pores. But it was quite important to say, without cardiolipin, not much happens. And because without oxidized lipids, happens also not so much. This was, and this is accelerating. So it's important that in our membrane, we always keep catiolipin when we do bugs. Here are the neutron data again. Now you see it's quite different with now for bugs. This is without bugs, and you see here bugs, how something is turning up here. And I have it on the next slide better, or the volume fraction. You see here now the bilayer, like before, fatty acid chain, no bugs, it's deuterated bugs. You add bugs, and then you see something is going in the bilayer a little. This is also this purple, but a lot of bugs is sitting outside and also take some lipids. So the bugs was surprisingly, which we still do not really know what happens, is making a hole in the bilayer, plus it removes the lipid to the surface. So this we discuss a little because we have supported bilayers, if it's also in free-floating bilayers like this, but this we will try next time with neutron time. But we thought it was quite surprising, and for the people at Harvard, it was a new mechanism that bugs can really shuffle out lipids because it needs a pore and puts them in a protein lipid complex outside the bilayer. So, and then we were doing some more stuff because by accident, when was COVID, we were not allowed to go to England. We only sent the sample to our colleague Luke, and sometimes he had time to play with this old machine a little. So he was doing time-dependent neutron measurements like kinetics, which is not so easy because you need a long time for the neutron signal. But he managed so from 15 to 100 and so on to look how bugs is going on the membrane and perforating and doing something. So he made analysis. You see quite nicely. You start before, and then bugs over a couple of hours is depositing more and more on the bilayer surface, which was very surprisingly and was more or less an accident because he saw something happens. Because often we do not see that because we add bugs and then we have 3 cells, and anyway the bugs is standing for a couple of hours before we measure. So this was by accident, so to say, he measured immediately something, we saw something happens, and it takes a while. So we were doing some kinetics, and we were a couple of weeks ago there. And before we were doing also FTIR on it. There's also there. In FTIR, you see the same behavior. You see also how the amide bond when bugs is added increases over time, that something happens with bugs and the membrane. You can plot it then. We thought, and if you plot it together, you more or less, just like the neutron peaks intensity, there's the amide peak. What you see finally at the end is we have a fast movement, which is around 10 minutes, which is probably protein adsorbing. And then you have a slow component, 100 to 300 minutes to 200, where it's perforating or going in and making pores. We thought on this, we made now a couple of measurements, and this works quite nice, that both agree. And what we are doing now, we looked at our catiolipin a little, because we wanted to see if catiolipin changes this. So we made membranes with small catiolipin amounts, and we made also some with a lot of catiolipin. And what you see quite nicely, if you have not much catiolipin, it takes much longer. And if you have a lot of catiolipin, it goes much faster. So it was clear the increase in catiolipin accelerates bugs insertion and lipid removal. And also what we analyze is that we use PC and catiolipin, that both lipids get removed, because we were not sure if probably catiolipin only gets removed, but we use deuterated lipids, as we also have deuterated PPC, and then you can see that both components get out. We're still working on this mechanism because we have not sorted it out yet. And if you plot now this intensity against each other, surface volume fraction, or how much bugs you have on the membrane, against time, you see the green one and the blue one is 15 and 5. So you have a different dependence. And the red one is a 10%, which does not fit perfectly in because it's from another session, which is not exactly the same condition bugs. Because our 10% we had in this series did not work. It was, I think it was too long standing. So it was bubbles on it. But you see the behavior similar. That they say, if you have a lot of catiolipin, it goes faster. If you have not much, it goes slower. But the endpoint probably are quite similar. Because after a while, bugs the same amount will have made enough pores and removed enough lipids. Okay, but now we have to think about a mechanism. And this was done also by Luke, because he looked a lot in literature and he made this model from our scattering length density profiles. This is like our profile with the fatty acid chains, the water, the heat groups. Before happening, here's the silica. That's why they're called supported bilayers. On one side, they're connected to the silica dioxide, so they're not really free floating. So we want to look for some free floating ones. If you add bugs, make a pore, and the protein is bugs, then has some proteoliposome or particles which is solubilizing or putting on the top. So to say it's like the lipids, bugs, and it's fitting from the distance exactly to our measurements. So here's the lipids and the bugs, and it fits quite nice. This is 100 angstrom above it. There's also some literature which confirmed a little this. It was from PNAS. It's a thought bugs or some parts of it can make pores which have not much protein. Okay, there was one, and the other was like Garcia. Yes, was said to say they used AFM on supported bilayers. As you could also see that there are some structures above the bilayer, around 40 angstroms, which are bugs clusters. Just like here's the figures. So in this 40, you can really think about it. If you say you go from the 60 to 100, or you go here from 75 to 125, that you have quite similar dimension for this. So it might be that this reflects a little what we see here with neutrons. Without this, we now analyze ready and then write it up at the moment, because we're doing a lot of different combination, different catalipin, deuterated bugs, non-deuterated bugs. But we still have also to continue further what happens next. Like I said, the holy grail is not this, it's not the BCL2. The holy grail is this. How does a complex look with bugs and BCL2 in your normal cell or mitochondrial membrane? If the cell is happy, we have this complex, and how does this big system looks like? And we have not done so many experiments, but we've done some, because some also failed. So what we are doing, we had now bilayers with BCL2 in the neutron work, and then we added bugs to it. And what you see quite nicely, bugs is doing something outside, BCL2 is still inside, but it's different to before. This one can see if one does with this more sophisticated analysis. Here is BCL2 in the membrane without bugs. And if you add bugs, you get more bugs in the membrane. You still get some bugs outside, but you do not get really lipid outside. So this means the bugs is interacting with the BCL2 quite nicely, but it makes no pores under these conditions. So we repeated this experiment once, but this failed, unfortunately, because something was aggregated. So we have to run it again with deuterated stuff. But then this was a quite clear indication or the proof of our hypothesis that BCL2 can prevent bugs from making pores. And under the condition we were doing it, it looks like that. But this was, we still have to analyze some data and perhaps still to repeat one or two of these experiments. But very nice that the bugs is behaving differently if it's seeing a membrane with BCL2 protein in it and not alone. Okay, now you can think about, okay, what else can we do? You can do a lot with these systems, very interesting. So, because NMR is our main method, we still want to get the structure out, so we can chop the BCL2 in smaller bits to look what other different domains are doing. You can also do neutron on it, but when we do NMR, so we can chop the BH4 on the loop, the binding pocket, the transmembrane part. So we can look at smaller parts of it, NMR, and then put them together in a big structure. And luckily, this works quite nice. If you say you look at this BCL2, two fractions, red and gray, if you add them together, you get more or less the black structure in. We thought, and this is quite nice, because as I say, you could determine the structure of smaller parts of the protein, put them together, and you get more or less the structure of the whole protein. So they're not unfolding. It's also shown here. We thought the only thing which is somewhere also made with the loops, and they look a little different. But then really you can look by making different parts of the protein, you can look over what the different domains are doing. So we thought that also we are working on it. Another thing what you can do, if you have the protein, you do not have to do neutrons on a lot of basic stuff. You can also go to do basic drug screening. Because we have a big national facility, we have big chemical compound libraries, and this we put BCL2 in the detergent in a tube, and then we're titrating fluorinated drugs against it with fluorine NMR. Then you can see quite nicely if something is binding or not. So you can look for fragments in the library, which are binders or hits or which not, and then you can synthesize and do more. So this is like also, we thought like a lot of drug companies do. They do screening against huge libraries. You can use NMR or other methods, but here we used fluorine NMR. We thought and published this like last year in Molecules, this strategy for it. What else can you do? We thought you can think about Sevenatoclax. I mentioned this is the only drug which is dissociating bugs away, BCL2, to kill membrane, but it works not only for lymphotic leukemia, unfortunately, as for all cancer it should. So Sevenatoclax is probably not too rigid to work for all kind of these binding pockets if something is a little different. We thought, and this is where companies look. They want to get something like this as a cancer drug, which is dissociating this complex away from each other, that bugs can go to mitochondria and make a pore and is not neutralized by BCL2. Because I think that around 50% of all cancers have some BCL2 involvement. We found in a lot of non-treatable cancers, they all have a big overexpression of BCL2. Then you can think about, okay, now we have this BCL2, what else can we do? We can do much more. We can also go to amyloidogenic diseases. And it is not that I come by accident to this kind of stuff, because I come originally from the Alzheimer field, protein aggregation on membranes with amyloid, A-beta, and so on. And superoxide dismutase is an amyotrophic lateral sclerosis. So it's more or less I'm slowly going back where I started many years ago. And there's a discussion that BCL2 can like be blocked by these amyloidogenic proteins. And if it's blocked, bugs is dissociated and can kill the cell. So a mechanism discussed in this ALS field that BCL2 can kill the mitochondria and the motor neurons. And what you see quite nicely, if you say you add it, it's a wild type APO, also a metal-free SOT variant, normally it's copper zinc in it. You see already, you see some changes. And this is also thought with people who have this ALS or Lou Gehrig disease called in US, that it's when the protein is not a holoprotein with the metals in, so if it's so half unfolded and so on, that it's really vulnerable to aggregation and become toxic. So in this, we work a lot on it. So we have different mutants. We have some mutants which don't show anything with BCL2. We have some others, so a lot. And the holo SOT, they do not show anything. So there has to be the APO state without metals to interact with the BCL2. Then we have another project still with synuclein in Parkinson, which you also see something if you put synuclein in, but you need a detergent concentration below creatine micelle concentration. If you have too many detergent or micelles or big micelles, then the protein cannot see each other. And you see also similar, you see weaker, you see other changes, something is binding. What it means, this we still have to figure out. It cannot be proteins like to bind each other, so we have to work on it. But at least we have two examples from these amyloidogenic diseases that BCL2 is interacting with them, which also supports some hypotheses that BCL2 might be also a key factor in these diseases by getting neutralized, and the apoptotic proteins can kill all these nice neuronal cells. Okay, now we can come to the end a little. So we say, who was doing all the work? There are a lot of people involved. There's from the group Jürgen Amek, who was postdoc, is just now moved to Texas, and Katan was a pharmacy student. Arthur, who's with BioNTech, Markus, he is now going to Uppsala, Martin, former PhD, is in Ilona, still here, she does a lot of metabolomics. Colleague Magnus does NMR, Tobias does our NMR facility, and then a lot of collaborators. Just like they helped a lot with some expression. Martin Schaka-Radek, they helped a lot with leakage. Jonathan's Chloe, they do SOT protein. Tommy Nylander in Lund and Hannah in Lund, they helped a lot with the Neutron work. And Mario, Luke helped, or especially Luke helped all with the Neutron works and in England. And Michael Hall, we do also cryo-EM on the BCL2 Bugs complex. Because then you need all funding a lot for all kind of things. And so we thought, and then I also have to thank my old boss. He more or less got me in contact with Luke when we were looking for people where we could do neutrons. There was a special Sweden, United Kingdom call. We thought, and I didn't know anybody in Havel. And so it was quite nice. And now I say thanks a lot to you all and hope you have some questions for me. Thanks.
Thank you very much for the very interesting lecture, Professor. And neutron reflectometry is a very new method for us. It allows us to do a lot of very elegant experiments. And please, dear participants, have you some questions?
I can switch off the shared screen so you can see me on the chat. The NMR signal we measure quite simple. You have to label. You make isotopes. You grow the protein or the E. coli in N15 medium ammonium chloride, and with carbon 13 you use carbon 13 glucose. So they get incorporated N15, and if you need also carbon 13. And for neutrons we also need deuterated. So often we only grow it in deuterated media, which you have to be careful because a lot of E. coli does not like it. So you have to adapt them a little to get a quite high deuteration degree. But the deuteration is important because neutrons, if they interact with deuterium much more than with protons. Okay, now I think if I saw it all quickly. Okay. With the herbal ones, we thought if you isolate, you can do it. You need the library. We thought you need like the specific compounds. They're different. There's waterloxy. You do not need fluorinated. You can also work with proton NMR to treat, and also like some people do, or the herbal or natural ingredients one, especially in some Pakistan. So this is not a big problem. Problem is to isolate the compounds you need and then to screen them.
And we also have a question, and does the same time is needed in alive cells for bugs formation of pores as compared with the time on lipid bilayers models?
It's similar. I think it's 10 to 20 hours apoptosis. So it's because Luke looked if it was realistic or not, but I think it's a slower process. You're not killing your cells in half a minute. It takes a couple of hours until they feel bad and then they say goodbye. It goes not quickly.
And what is your opinion? Is this process, what forms of cancer are most perspective for being treated with such methods of such killing?
No, we thought Abbott or AbbVie with this kind of venetoclax, they had your hope they have a drug which would help a lot of cancers. But this chronic leukemia, and perhaps there's a side drug for other treatments. So the general mechanism or strategy is probably to get a more universal drug which is binding with higher affinity. And the other problem is that a lot of cancers have BCL2 as a main problem, but often they also overexpress the other cell protecting proteins, BCL-XL. So this might also sometimes, if you stop BCL2 from working, that the others can be overexpressed. So it's not so a simple mechanism that only one BCL2, you stop it and it's enough. This is also that cancer is quite heterogeneous, but then in half the cases, I think BCL2 is involved. So I think especially I work a lot with prostate cancer, metabolomics. All who have like get prostate cancer back after a couple of years treatment, or they have like in the final stage, they all have high BCL2 overexpression levels. So at least if they get incurable and so on, that they all produce a lot BCL2. Perhaps this might be a good way perhaps to treat this often not incurable one. Find a better drug to stop this BCL2 function. So I think it's a holy grail. The pharmaceutical industry puts a lot of effort in because this is a fundamental mechanism where you can switch off cancer cells, but they have not so many drugs on it. So there's Abbott, has this venetoclax, but there's not so many others. Sometimes they use a combination, but it's not a straightforward thing. If not, they would already make a lot of money with it. Mm-hmm.
Thank you very much. Dear participants, do you have any other questions? Now we have a lot of participants, more than 120.
That's good. Good.
Yes.
I couldn't remember how many you had last year. Was it also over 100 or how much was it or more?
Smaller. We had smaller.
Smaller. I think it was smaller. It was not so much. Yeah. No, then I say thank you a lot. And I think I just met my colleague in Solomon before lunch. He said you have him on Friday or on the 1st. Yeah.
Oh, thank you very much for your kind agreement and for your very interesting lecture. Thank you. Have a nice day.
And have a good week, you all. Bye-bye.